Welcome back to this next video on the Gregor Mendel. In the first part of this video, I've told you that Mendel, he was born in 1822 and he was a scientist. He was an Augustinian friar and he remained the abbot of the St. Thomas. What Mendel did was that joining the St. Thomas Church, he went to the University of Vienna in 1851 by the then abbot, the Abbot Nab, and in the University of Vienna he remained under the influence of three important teachers, the Unger, who was a plant taxonomist, the Doppler, who was a physicist, and the Eddinghausen, who was actually a mathematician. And especially the Unger and the Eddinghausen, they have a very prominent effect on the laws of the Mendel. What Mendel did was that he started working on the blending inheritance, and I've told you that the reason of his success was that he chose his experimental material very wisely. Most of the scientists, they were working in that particular time on the blending inheritance, but as Mendel chose his experimental material very wisely, he was able to deduce the results that we see these days. Mendel, he actually worked with the pure breeding varieties, and I've told you that he chose 7 traits to work with. One of the traits that was the seed shape, and the seed shape that come in 2 varieties, like the seed shape can be round or the seed shape that can be wrinkled. Similarly, he selected 7 traits that he studied in the pure breeding varieties. What Mendel did was that one of the key to his success was that he started work on the monohybrid crosses, because if you're dealing with a single trait, you will be able to deduce the results very good. But if you are working on a variety of the trait at a single time, the result that will be very complex and you will not be able to deduce anything. So what Mendel did was that in one of his experiments, he crossed the pure breeding homozygous tall plant to a pure breeding dwarf plant. And he did the cross fertilization, and in the last video I've told you how he did that in the both direction. So his first experiment in the monohybrid crosses was that he crossed a homozygous tall plant, a pure breeding tall plant. Now we will say that is homozygous at both loci, and both of the alleles in this pair, they will be the same, they will be identical to each other. So he crossed a tall plant having a genotype of the capital T to a dwarf plant having a genotype of the small t and the small t. And what he got in the F1 generation was that all of the plants, they were tall in nature, and from here he deduced that the tall trait that is dominant and the dwarf trait that is a recessive one. What Mendel did next was that he went for the self-fertilization, and in the self-fertilization what he did was that he crossed the F1 with the F1. That means the tall plant that he got in the F1 generation, he actually allowed them for the self-fertilization, and by doing that, when he got his F2 generation, he deduced that the ratio of the tall to dwarf, that was 3:1. That means that for every 3 tall plants you will be getting 1 dwarf plant, so the ratio was actually 3:1. And what he see was that the phenotypic ratio of the tall to the dwarf, that was actually 3:1, but the genotypic ratio, that was actually 1:2:1. What Mendel did was that by seeing this, he said that the tall plant that we are getting in the F2 generation, what Mendel noted was that the plant that expressed the dominant trait in the F2 generation, that can either be of the parental type. By the parental type, he means that the tall plant that you are getting in the F2 generation, that will be homozygous in nature. That means that both of the alleles, they will be like the capital T and the capital T. The other tall plants that you are getting in the F2 generation, that were actually the hybrid type. By that he means that the genotype of this particular plant, that was the capital T and the small t, because in the last video I've told you that Mendel went for the F3 generation as well, and in the F3 generation what he got was that the F2 tall plants, when they were allowed for the self-fertilization, they actually produced 2 types of the categories. some of the tall plants that they produced only the tall plants, but some of the tall plants of the F2 they produced a mixture of the tall and the dwarf plants. From here, the Mendel said that the tall plant that we are getting in the F2 generation, they are actually the true breeding dominant. Like this one is actually referring to the true breeding dominant. This means that if you are going to cross them, like this will be a true breeding one. That means that if this is homozygous, that means that all of the gametes that you will be getting, they will be having the capital T allele. And if you are going for the fertilization, all of the time you will be getting the tall plants, and that means that you are talking actually about true breeding tall plant, or you are talking about homozygous tall plant. So what he said was that in the F2 generation we are getting this one is actually referring to the true breeding dominant. This 2 is actually referring to the non-true breeding dominant. You are talking about a dominant trait, but you are actually talking about a non-true breeding dominant. That means that if you allow for the self-fertilization of these plants, they will be actually producing the tall plant as well as the dwarf plant. This is what you can actually represent like here, that you will be having the gametes containing the capital T allele, you will be having the gametes containing the small t alleles. So if you are going to cross them, you will be getting like this will be a tall plant, this will be a tall plant, this is a tall plant, but this is a dwarf plant. So you will be actually getting a mixture of the tall and the dwarf plant. So Mendel deduced that the phenotypic ratio that you are getting, like 3:1 phenotypic ratio, but if you talk about the genotypic ratio, this 3:1 can actually be divided into a 1:2:1 genotypic ratio. Now what he did next was that he as I've told you, that he presented his principle of the dominance, that if in a heterozygote one allele that may conceal the presence of the of another. We have already discussed that. And he talked about the principle of segregation, that during the gamete formation, each member of a gene pair has an equal probability of being in a gamete, and a gamete gets only one. After doing work with the monohybrid crosses, Mendel also worked with the dihybrid crosses. What I mean by dihybrid crosses is that this di actually refers to two. That means you are now focusing on two traits instead of a single trait. The hybrid means that both of the traits that you are focusing on, they contain two different alleles. In a cross means that actually you are actually crossing two parents and they are combining their genotypes. One of the Mendel cross was that he focused on two traits in a single cross. And the two traits that he chose was, one was the seed color and one was the seed shape. So now you are talking about two traits. One is the color of the seed, the other one is shape of the seed. If you talk about the seed color come in two varieties. The seed can have yellow colors, the seed can have green colors. This yellow color of the seed, that is a dominant trait. This green color of the seed, that is a recessive trait. When Mendel was doing his monohybrid cross, he actually deduced all of the 7 traits that he observed, that which one was the dominant and which one was the recessive one. So as far as the color of the seed is concerned, the yellow color, that is a dominant trait, therefore we represent that with a capital Y. The green color of the seed, that is a recessive trait, therefore we represent that with a small y, because this is accustomed to represent the dominant allele with a capital letter and the recessive allele with a small letter. The seed shape, that also come in two varieties. The seed can be round in shape, and this round shape is dominant, therefore we represent that with a capital R. The seed can also be wrinkled in shape, and this wrinkled shape, that is a recessive trait, therefore the allele for it is represented by the small r. What he did was that in first cross, as I've told you, he actually worked with the true breeding varieties. So in the first state of the dihybrid crosses, what he did was that he crossed the homozygous round yellow plant to a homozygous wrinkled green plant. So if you talk about the homozygous round, that means that this particular capital R and capital R, they are identical to each other and they are homozygous in nature. And if you talk about the color of the seed, that was yellow, that means that it was homozygous in nature. So the pair will be having the capital Y and the capital Y. He crossed that with homozygous wrinkled green. That means that the alleles for the shape, that will be small r and small r, and the alleles for the color will be the small y and the small y. What he did was that when he crossed them in the F1 generation, what he got was that all of the plants, they were actually having round yellow in nature. All of the plant that he got in the F1 generation, when he crossed the homozygous round yellow to the homozygous wrinkled green, all of the F1 generation plants that were round yellow in nature. Now when you talk about the dihybrid crosses, the most important thing is that you should know how to make the gametes. Now if you talk about this one, this is the capital R and this is the capital R. This is a pair of the allele and it is representing only one trait. And this capital R, it is actually representing the shape of the seed. Similarly, this capital Y and capital Y, this is showing the other trait, and this is actually showing you the color of the seed. And as he said in the principle of segregation, that during the gamete formation, the members of a pair, they get separated from each other, and during the gamete formation, one gamete is actually going to get only a single member from this pair. So if you talk about the gametes of this particular parent, what you can see is that this capital R from this pair, it can actually combine with this capital Y from the other pair. Similarly, as the probability is equal, this capital R can also combine with the capital Y, the second capital Y of this pair. This capital R, the second R of this pair, it can actually combine with the first member of this pair, but it can also combine with the second pair of the second member of this pair. So what you can expect from the first parent is that the gamete that you can get, you are only getting a single kind of the gamete, which will be the capital R and the capital Y, because both of the traits, they are in their homozygous form. Same will be true for the second plant. This pair of this pair, the members, they are actually getting separated from each other during the gamete formation. The member of this pair, they will be getting separated from each other during the gamete formation. The first small r, it can actually combine with the first small y, but this first small r, that can also combine with the second small y. Same will be true for the second r. The second r, it can combine with the first y. This small r, it can actually combine with the second small y. So this particular parent, again, it is going to produce only a single kind of the gamete, that will be small r and the small y, and this parent will be producing the gamete having capital R and the capital Y. In the F1 generation, if you are going to cross them, what you actually see is that this parent, this side of the Punnett square is showing you the gametes of one parent. We are representing this for the homozygous round yellow, so we are representing that with the capital R and the capital Y. The top side of the Punnett square, it is actually showing you the gametes of the second parent, and as the second parent is producing the gametes, the small r and the small y all of the time, therefore we actually write that on the top of the Punnett square. If you cross them, you will be having this gamete can combine with this gamete, giving you an offspring with a genotype of capital R and the small r, capital Y and the small y. If you combine this gamete with this gamete, again you will be getting the same genotype, the capital R and the small r, the capital Y and the small y. If you do that for all of the 16 boxes shown over here, all of the time you will be getting only a single kind of the genotype, and hence a single kind of the offspring. So therefore it is not needed to repeat that. If you see that both of the parents, they are producing a single type of the gamete, crossing them once is enough to show you the results, because if you do that, that is going to waste your time and you are actually getting the same kind of the results. So in the dihybrid crosses, when he crossed the homozygous round yellow plant with a homozygous wrinkled green plant in the F1 generation. All of the offspring, they were round yellow, but of course their genotype, that was heterozygous, because this capital R is dominant over the small r, so the phenotype for this particular trait will be round, as the capital Y that is dominant over the small y, so the phenotype that will be yellow in nature. What he did next was that he allowed the self-fertilization of the offspring that he got in the F1 generation, as he did in the monohybrid crosses. He followed the same pattern in the dihybrid crosses as well. In the F1 generation, all of the plants that were actually heterozygous round yellow. By heterozygous, I mean that if you look at this particular pair, this is actually showing you the shape of the seed, which is actually the round one. Now, if you look at both of the members, they are different from each other. Therefore, we are saying that this particular plant is heterozygous for the shape of the seed. Similarly, if you talk about this capital Y and the small y, both of the members of this pair, they are different from each other. But as the capital Y that is dominant over the small y, therefore the phenotype is yellow, and we say that it is heterozygous for the yellow color. And of course the second parent, that was identical to because you are crossing the F1 to the F1. If you talk about the gamete formation over here, this capital R, it can combine with this capital Y, giving you a gamete, the capital R and the capital Y. But at the same time, this capital R have equal probability to combine with the small y, giving you another kind of the gamete, which is the capital R and the small y. Similarly, this second small r, it can actually combine with the capital Y, giving you a gamete of the capital R, the small r and the capital Y. But the small r can also combine with the small y, giving you a gamete of the small r and the small y. Now you are actually getting 4 different kind of the gametes. As the other parent is identical to the first parent, again you will be actually getting 4 kind of the gametes for the second parent as well. Now what he did was that when he checked the offspring that he got in the F2 generation, he actually got 4 different kind of the offspring. There were round yellow offsprings. There were round green offsprings, there were wrinkled yellow offsprings, and there were wrinkled green offspring, actually with a ratio of 9 to 3 to 3 to 1. These round yellow, they were 9, the round green, they were 3, the wrinkled yellow, they were 3, and the wrinkled green, there was only 1 offspring, which were actually producing a ratio of 9 to 3 to 3 to 1. Now, how this work? How the heterozygous round yellow plant, when you cross it with the heterozygous round yellow plant, how you are actually getting to going to get the 9 to 3 to 3 to 1 ratio. I have explained that in my another video on the Punnett Square, and I will share the link in the description. So what he did in the dihybrid cross, he actually got a ratio of 9 to 3 to 3 to 1. Now if you look at this 9 to 3 to 3 to 1 ratio, and if you are going to convert that into the monohybrid crosses, because the first thing that Mendel was doing, that he was actually getting a ratio of 3 to 1 for the monohybrid crosses. So if you split this, let me split this into the monohybrid crosses. Like in one of the traits that we are focusing on is the shape of the seed, and the shape of the seed, that can be round or that can be wrinkled. 2 type of the shapes you can expect. Now if you look at the shape over here, like you are actually getting round, 9 round over here, and you are actually getting 3 round over here. So you are actually getting a total of 12 round over here. Now if you talk about the wrinkled one, you are getting the 3 wrinkled over here, 3 wrinkled over here, and 1 wrinkled over here. So you are actually getting 4 wrinkled over here. So if you simplify this, again, you are actually getting to get a ratio of 3:1. If you talk about the color of the seed, talk about the color one, again the color that was yellow, or it can be green. So if you count the yellow one over here, 9 yellow over here and 3 yellow over here, so you are actually getting 12 yellow over here. And if you talk about the green one, you have got 1 green over here and you have got 3 green over here, making a total of 4. So if you are going to split them, again you will be getting a ratio of 3:1. That is what Mendel got in the monohybrid crosses. So if you split this and there's a dihybrid cross into the monohybrid cross, again you are going to get the ratio of 3:1, which actually proves that the monohybrid crosses that Mendel did, that was okay, or the dihybrid crosses that Mendel did, that was very okay. Based on the results of these experiments that he got in the dihybrid crosses, he actually explained, he actually gave his his third principle, you can say, and which is actually known as the principle of the independent assortment. Now what this principle of the independent assortment states that during the gamete formation, the allele of different genes, now as you are seeing that we are focusing on 2 traits, so we are actually talking about different genes. So during the gamete formation, the allele of the different genes, they segregate or assort independently of each other. Like if you apply this on the cross that we just discussed, that the color of the seed, the gene for the color of the seed, that has nothing to do, or that has no impact on the genes which is responsible for the shape of the seed. So this was his third principle, which is known as the principle of the independent assortment, that during the gamete formation, the allele of different genes, they are going to segregate, assort independently of each other. Now this principle, of course, exceptions are there when you talk about the genes that are linked to each other, but at that particular time he was not talking about the linkage, but what he observed was that during the gamete formation, the allele of the different genes, they are actually segregating or assorting independently of each other. But when Mendel he got these results, so he presented the results to the Abbot Knapp, and the Abbot Knapp wanted Mendel to present his work to the Philosophical Society of Natural History of Brno. This was a society which was actually holding different kind of the seminars in which the scientists, the local scientists, as well as the scientists outside of the Brno, they were actually presenting their work. So Mendel, he was given the date of February 21, 1865 to present his work at the Philosophical Society of the Natural History of Brno. The Philosophical Society of the Natural History of Brno, when they held these seminars, they actually divided the seminar in 2 sessions. In the first session, someone outside the Brno, he was actually presenting his work, and then in the second session, the scientists of the Brno, they were actually presenting their work. In February 21, 1865, in the first session, the book of the Darwin, that the evolution, that was on the evolution, the name of the book was Origin of the Species. That book was presented, and as that book at that particular time was a controversial book, and if you are presenting that, some of the people, they were happy with the book, some of the people, they were not happy in the book. And in the second session, when the Mendel has to present his work, no one came back to listen to the presentation of the Mendel. So the presentation of the Mendel, that was set for the next meeting, that was on March 8, 1865. What Mendel did in his presentation was a disaster. When he looked at the people that were sitting in front of him, he got nervous. And he was actually a patient of the melancholia, or you can call at this particular time that he was a patient of the depression. So he started his presentation, and he started saying that I went for the monohybrid crosses, I got a ratio of 3:1, then I divided that into 1:2:1, then I got a ratio of 9:3 to 3:1. Not even a single one got what he was saying about. So he presented his work, disastrous presentation, but what he did was that he published his work, and he got 40 prints from his paper. No one get anything from his presentation, but what he did was that but a good thing he did was that he published his work, and he got 40 prints out of the paper, and he sent them to the leading scientists of those particular time. One of the leading scientists of those particular time, that was Carl von Nageli. He was actually a botanist, so as Mendel he was working with plants, so the paper, it was also sent to Carl von Nageli. When Carl von Nageli he studied the paper of the Mendel, he said that I'm not going to believe your data unless you you prove your work in the plant which is known as the Hyraceum. Now Hyraceum, it was the plant that Carl von Nageli he was working with. So he said that I will accept your work. Carl von Nageli told Mendel that I will accept your work if you are going to prove your result in the Hyraceum. Mendel started working on the Hyraceum, and he published his next paper on how he was not able to produce his result in the Hyraceum plant. So as he was not able to produce his result in the Hyraceum plant, his work was not accepted by the Carl von Nageli, and his paper, the Mendel of the paper, that was set aside, and no one was able to believe the results that he got. Now we know that why Mendel was not able to achieve his result in the Hyraceum plant, because the Hyraceum plant, they produce mitotic flowers. They do not produce the meiotic flowers. And as Mendel he was working with the gametes, and if a plant is the flower of the plant, they are mitotic in nature, of course you are not going to get any results for any results that Mendel got in his pea plant. In 1867 he was made the abbot of the monastery, and whether he discovered the linkage or he didn't discover the linkage, we do not actually know about this, because after Mendel died, this person, the Anselm Rambusek, he was made the abbot of the monastery, abbot of the church, and what he did was that he burnt all of the library of the Mendel. So we do not know what Mendel did after 1867, because all of his work that was burned by the Anselm Rambusek. His work was rediscovered in the early 1900s by three researchers, the Erich von Schremark, the Hugo de Vries, and Carl Collins. And all of them, they claimed the work from themselves. All of them said that this is our work. But when they observed that we are referring to the same work, all of us, we are referring to the work of the Mendel. After then, the credit that was given to Mendel, that yes, this particular work, that was the work of the Mendel, and that was not our work. So this is how his work that was rediscovered.